Today, we're going to crack open the case file on one of the greatest scientific detective stories of all time, the discovery of DNA. Seriously, it's a tale of mystery, intense rivalry, and a breakthrough that completely changed our understanding of life itself.
So think about this for a second. For the better part of a century, this was the central question that had scientists totally stumped. I mean, they knew something inside our cells was passing traits from parent to child, but they had no idea what it was or how in the world it worked. It was like hunting for a ghost in the machine.
Before scientists could even dream of reading the Book of Life, they first had to find it. The search was on for the actual physical molecule responsible for heredity, the blueprint of life.
And let me tell you, this was not a quick search. It was a long, really challenging investigation, just full of false leads and dead ends. Everyone was desperately looking for that one single substance that could somehow hold all the complex instructions for building a living thing.
All right, so this brings us to our first big mystery, identifying the culprit. Which molecule was passing traits from one generation to the next? It turns out there were a couple of key suspects. You can see here the case was built piece by piece over more than 80 years. It kicked off with a chance discovery, was followed by some really puzzling observations, and finally, it all came down to one definitive experiment. This was a slow burn, not some sudden flash of insight.
Believe it or not, the very first clue in this whole story was found by Friedrich Miescher in used pus-soaked surgical bandages. Yeah, you heard that right. He isolated this weird stuff he called nuclein, but neither he nor anyone else had any idea how important it was. For decades, DNA was literally hiding in plain sight.
For a long time, the scientific community was basically backing the wrong horse. Proteins were the prime suspect. And you can kind of see why, right? Life is incredibly complex, so the molecule carrying its instructions must also be complex. Proteins, with their 20 different building blocks, seemed perfect for the job. DNA, on the other hand, with only 4, it was just seen as way too simple to be the master molecule of life.
Well, the debate was finally put to rest in 1952. Alfred Hershey and Martha Chase pulled off this brilliant experiment using viruses and radioactive tags. They proved, without a shadow of a doubt, that it was DNA, not protein, that viruses injected into bacteria to hijack them. And with that, case number 1 was closed. DNA was our culprit.
But you know how it is in science? Solving one mystery just opens up another, even bigger one. We knew DNA was the stuff of life, but what did it actually look like? Its structure had to hold the key to how it stored and copied information. So the race for the structure was on.
And boy, was it an intense race. You basically had two key teams going head-to-head. In London at King's College, you had Rosalind Franklin and Maurice Wilkins, who were the experts at collecting the hard data. And then over in Cambridge, you had James Watson and Francis Crick, who were all about building theoretical models to see what might fit the evidence.
Now we have to talk about Rosalind Franklin, because her role in this cannot be overstated. She was an absolute master of a really difficult technique called X-ray crystallography, which is basically a way of using X-rays to see the shadow a molecule casts. Her work was meticulous, methodical, and it produced the single most important piece of evidence in this entire investigation.
And this is it. Photograph 51. To you and me, it might not look like much, just a blurry X. But to a crystallographer, this was the smoking gun. That X-shaped pattern screamed Helix. It was the visual proof that DNA had a spiral structure. Okay, so this is it, the final act. All the clues were finally on the table. You had the chemical rules, you had the photographic evidence, and you had the bold theoretical work. It was time to put the puzzle together once and for all.
This is where Watson and Crick really shine. They were like master puzzle solvers. They took Franklin's data, which had been shared with them, by the way, without her direct knowledge, and they combined it with another critical clue from a chemist named Irvin Chargaff. He'd figured out that in DNA, the amount of A always equaled the amount of T, and C always equaled G.
So, using all this information, they started building their model. The best way to think of it is like a twisted ladder. The 2 long strands are the sides of the ladder, made of this sugar phosphate backbone. And the rungs? Those are the base pairs. A always hooking up with T, and C always with G, holding the whole thing together, and crucially, carrying the genetic code.
And there it was, the double helix. The sheer beauty of this model, which they proposed in 1953, was that its structure immediately revealed its function. You could instantly see how the strands could just unwind, and how each side could serve as a perfect template to copy itself. It was the elegant answer everyone had been searching for.
But the discovery of the double helix wasn't the end of the story. Not even close. It was the first page of a whole new book, the start of a DNA revolution that would completely transform our world.
In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize for the discovery. Rosalind Franklin, whose photograph 51 was so pivotal, had tragically died 4 years earlier, and the Nobel isn't awarded posthumously. Today, her absolutely essential contribution is rightly so, widely and deservedly recognized.
The legacy of this discovery is all around us. I mean, it is the foundation for everything from genetic engineering and the massive Human Genome Project to the DNA evidence used in modern forensics and the personalized medicine that is shaping the future of healthcare.
So, when you look at the whole story, cracking life's code wasn't one person's single eureka moment. It was a cumulative triumph, built on the hard work of dozens of scientists over decades. Each one sort of passed the baton to the next, adding another crucial piece to the puzzle.
This whole story was like a great scientific relay race that spanned generations, and it leaves us with a final question to think about. What's the next great scientific baton, and who out there is getting ready to carry it forward?